Well, welcome everybody. So I'm here to talk about my story through STEM. Can everyone hear me? Awesome. So it's not at all straightforward. You'll learn that through the stories I'll be telling. I won't go into too much detail, but very specifics of my research or the illustration, because I want all of you to leave this seminar with some way to succeed in STEM and tidbits that people in research don't really talk about, or how to be a good worker, for instance. And so a little instance like that I can talk about. But one thing I want to start with was a recent art residency that I did up in Crater Lake National Park in Oregon. And so we've been telling the story, but it's kind of funny how this situation happened where once I was accepted this residency, I was with a bunch of the national park rangers, and we left the national park to kind of observe where Crater Lake used to be. And Crater Lake used to be a huge volcano that exploded a few thousand years ago, leaving this now crater or caldera that's now just full of rainwater. And so we were an hour and a half outside the park, and we're looking at this little podium describing what Crater Lake used to be like. It used to be an entire volcano. And someone smells, and there were a couple of rangers and another art resident, and one of them was like, something smells awful, it smells terrible here. And we look on the other side of the podium, and there's a petrified coyote specimen right on the other side, that we're all looking at it, and we could tell through its scattered skull, shattered skull, I can talk, that I think it was shot. I think it was someone's farmland. And so everyone's completely grossed out, but me working at a natural history museum, and I was like, ooh, that's a really cool specimen, can I collect it? And so, word of wisdom, don't collect it in a national park, because it's completely illegal. And so we were out of the park, and we were on a highway, we weren't in any kind of park, so I asked the national park rangers, can I collect the specimen for educational purposes and to illustrate? And they were like, you can come back, but you can't put it in a government vehicle. It's like, all right, well, I'll go back to my cabin that I was staying in, picked up my stuff, got a snack, drove to the store about an hour away, and a hardware store, and I bought vodka for sanitizing purposes. I bought gloves, I got a bucket, I got knives, and hand sanitizer. And I went to the register, the guy didn't ask any questions, pretty weird. And so I got all that stuff, and I got soap too, because I had to clean the specimen. And so I go back to the specimen, and I see this perfectly preserved coyote specimen. There's basically no meat on it, it's just bones that are just sun-dried. And so I collect the skull, and there's insect crawling out of it because they're digesting the rest of the meat. And this isn't the grossest story that I'll tell during this presentation, so just a heads up. And so I collect the skull and part of the vertebra. You'll learn why I'm into this kind of stuff. And so I put it all in a bucket, I drive back to the cabin. Thankfully I'm staying in the cabin by myself for these 2 weeks. And so I immediately start boiling water. I put salt and soap in the bucket full of boiling water, and I put the coyote specimen in there. I have my phone kind of timing how much I'm going to boil it over to get the meat and the fur off this specimen. And so I'm talking about a boiling coyote skull. If none of you have done it after this, you probably won't want to. And so I'm taking care of the skull in the bucket, and then all these insects that were in the skull that are now scattered all over the bathroom, kind of freaking out. Oh great, there's all these things scattered in this bathroom that I'm staying at for 2 weeks. I gotta go clean everything up. And so, and then kind of on the counter, I have my gloves on because I don't want to touch the specimen, obviously. And so I move my phone, it immediately starts ringing when my friend starts calling, but the volume's all the way up, so it starts freaking out. And I hit my elbow on the side of the sink. Immediately the vibrations go to the bucket, and I don't know why this happened, but the coyote skull jumps out of the bucket because there was still bubbles and air in its skull, and so it kind of shook out of what was keeping it down. It started popping out at me, and my gut reaction, everyone's gut reaction is to be just shut it down. And so I kind of forgot until seconds above the water that there was boiling water, and so I stuck my hand in there to push the coyote skeleton back in the water. And seconds later, I'm oh, it's boiling water. It's burning again. And I'm at that point where I'm what have I done to get here to this point? Everything's just happening. I took this specimen because I'm going to illustrate it. I got the necessary equipment because of my background in infectious diseases. And then I knew how to deal with this skeleton because of my work with the whale necropsies I'll tell you all about. And so I kind of got to this point where I'm like, how did I get to this point as an artist and researcher, as a scientist? And things are coming together in this way. This is pretty funny, kind of hurts, but hopefully you guys will have more positive experience because things do come together at the end. It doesn't have to happen with you burning your hand off. But overall, it's a beautiful specimen we have looking at it. And so, if you can see this Venn diagram, it's basically not a Venn diagram because the more I do research and the more I do art, the more I realize that they're both the same. And that you both have to be a creative individual or be creative to kind of tackle a scientific protocol or how you're going to go about illustrating something and drawing that for communication purposes. And so they're both very successful methods, as we know, to explore the natural world. And we'll kind of talk about the intersection of both of these, but also the differences between art and science and how you can understand the world in a more complex way. And so here are my parents, and it's me. I teach classes in Yosemite every once in a while. And it's just a cute photo of my parents. And like what I like to talk about is mainly my dad is a neuroscientist and a kinesiologist. My mom is an artist, an art historian, and I like to put that, still trying to figure things out, because even though I'm up here, and I do research at the California Academy of Sciences and illustration, there's still a lot in STEM that I'm still trying to figure out as a researcher, as an incoming scientist, and I'm trying to relay all this information to all of you, so that when you do graduate or you go on to your next either program or job, you'll have a better idea of knowing that people that you work with are still trying to figure things out too. And so, how many people here have gone to the California Academy of Sciences? Awesome. So, a few stories that I have, I'll tell in a little bit, but it's a pretty entertaining place. For those of you that have been there, if you haven't, I highly recommend it. We were founded in 1853, only 3 years after California became a state. So it's been around for a very long time. And it was founded as a research institution, only for researchers and natural historians to go around California, this new state, and document wildlife plants and animals, and to collect it, and to keep it in a museum setting for people in the future to research. And so here at the museum, we have this now modern take on natural history called the Institute of Biodiversity, Science, and Sustainability. And so the ones in orange in botany, genomics, entomology, and microbiology, those are the departments that I work in. Every day is a little bit different. I'll kind of tell you my timeline of how I got into each of these departments because it's not at all straightforward. And so these are my titles here at the museum: scientific illustrator, infectious disease biologist, a science communicator, storyteller, and a whale forensic researcher. And so there's a lot of stuff going on in the museum. There's many more opportunities than I'll actually be able to discuss here. So if you have any questions afterwards, feel free to come up and ask me any questions, or even after the presentation, or email me too, if you don't have the chance to. And so does everyone have a pencil or paper? Okay, I thought about noise. Does anyone take notes anymore in lectures? But what I'd like you to do, at least for the next minute or two, is think about what you had for breakfast or lunch or something you ate today. And do like a very quick doodle of some of the ingredients that may have been in that sandwich or the breakfast bar. Or if you had coffee, you could draw like a cup of coffee. Okay?
I think just ate bacon.
What was that?
I think just ate bacon.
You could draw bacon too. Just like little squiggly lines of bacon. No pressure. You don't have to draw. I don't know what you said at all. It's not the point. It's exercise. If you don't have a pencil or paper, you think about what you might draw out to. Just visualizing things in a way. you're probably looking at your drawing, I do this all the time too, where you probably look at it, you're like, oh my god, it's not what you expected, or it's just, you visualize something, and it's pretty difficult to translate that onto paper. That's normal. But now for this next one, share your drawings with the person next to you. Or just say that and then you draw something. But tell them what ingredients you need for this next. And so we can go back as to why each of us illustrated one of those. We can go on about that for literally days. If you really think about why we just illustrated some of those subjects, we'll kind of cover at the end of it the overarching theme of why we just did that exercise. But ultimately, you think about why did you just draw all that, and was it exactly what you imagined, like when you thought of it in your head? You thought about like spinning your copy and try to translate that onto paper. Were you able to effectively draw that in a way to communicate it to someone else? And so a lot of these illustrations don't need us to have a wording behind it, so we can just have these drawings to communicate these concepts ourselves. And so that's kind of the use of scientific illustration and the use of language that we've developed over thousands of years. And so it's a pretty useful tool. Speaking about it isn't really an option. And so that is one of the reasons why I have found myself in STEM, is that I found a way to do research using illustration and to do artwork via a scientific concept. So using the boat to help the other. And one of the main takeaways from this presentation, as I mentioned before, is that the pathway to STEM is anything but straightforward. You are often looking at a CV. This is my very basic CV that I have right now, but you kind of look at this, you don't really know the person behind it. And you see this person up here giving you a lecture with your professor, but you really don't know like the intimate details of how they got to that point. And so I'll kind of share with you my story of how I got to each of these points, who I met along the way, and how that helped influence me to get where I am today, and different options you could take in STEM too. And so I started off at UC Santa Cruz, studied botany and evolutionary biology, and afterwards I immediately went into scientific illustration graduate program at UC Monterey Bay. And then after a few years of not going to school, I had to take a break from it. And I found myself at the California Academy of Sciences, and then through San Francisco State University. And so in undergraduate studies, I didn't really know what to focus on at first. I'll tell you another story at the end of this lecture as to why I started studying sciences in college. But I found it is the most useful way for me to get to know a bunch of different fields and to use undergraduate to kind of just spread everything out and see what I wanted to focus on. And I did, after undergraduate, I still didn't even know what I wanted to focus on. I wanted to study everything after all these biology classes, which were helpful. But in terms of the next degree or next step, it was like, what did exactly I want to do? And you don't really know that after undergraduate. But I did know that one of the best ways for me to learn something in class, any class, was if it was photosynthesis, kind of drawing it out, or getting that chapter, like the very detailed paragraph in your plant physiology class, for instance, and kind of translating that paragraph into a drawing, or kind of using the illustration that you have in your textbooks, the scientific illustrations, and redrawing that. So that was the best way for me to feel a very visual learner. And I can't go through an entire book or a whole chapter with just text because I'm not really going to pay attention. It's not really that. It's not the best way for me to absorb information. But I did find that redrawing these and drawing out these paragraphs was the best option for me to retain this information even after an exam. Because exams are obviously pretty important. And also, my favorite classes, and my favorite professor, because she's just so ecstatic, just so happy about talking about marine mammals all the time. She's like jumping on desks and talking to us about dolphins and like dolphin respiratory systems and whatnot. But it's that kind of person. It's like, this person's so excited about what they do, and I love it, and it kind of sticks with you. You have a very passionate person that talks about what they study and why. And so afterwards, after undergraduate, I went into the scientific illustration program at CSU Monterey Bay, and I look at the portfolio now that I submitted to them, thinking, I was like, I'm so good. It's awful. And like, I look at the sketches that I did, I'm like, whoa, I can't believe I sent that. But what I did do during the undergraduate program is go out of my way to meet the people in the program and to talk to them about what the program is about, if they offer any summer classes for you to practice, and also what they recommended I do as someone that doesn't really have that much of an art background, and how I can succeed to be a scientific illustrator. And so during this program, I focused on a lot of tropical biodiversity and conservation work, really just illustrating species that I found in the tropics, because that's just something that has been like a huge passion of mine since the get-go. But one of the things about this program, it's like a 1-year intensive program where it's a boot camp. They kind of like whip you into shape and have you a professional scientific illustrator outside of the program. And they have you do a couple of internships of your choosing. And I was connected with one of the internships. There's someone that I volunteered with in undergrad who just came out from Madagascar, and she was like, she emailed my advisor in undergrad. She was like, I need someone to basically crush up plants that I collected in Madagascar in liquid nitrogen and just be in a lab for hours and like crushing plants in like a mortar and pestle. She didn't want to do it. She was like hired professor, so she doesn't at the time. But she had an undergraduate, which is me, and I, after probably 4 years later, after I did the illustration program, I emailed her, I was like, hey, do you know of anyone that might need scientific illustration work in Madagascar? And she emailed me a couple days later. She was like, actually, the advisor, the main executive director of this research station in Madagascar needs an illustrator. Like, are you free to fly out in a couple days? Thankfully, it was at the end of the program, so I was like, yeah, sure, I'm out. And so I flew to Madagascar, and I was there for about 5 months to illustrate lemurs, moths, and birds in the national park. At first, and this is why I say plan ahead and learn how to work independently, because when I was there, I was hired by the National Park of Madagascar, and I had an interpreter to talk to me, and like just back and forth because I didn't understand Malagasy. And so when I got there, like I said, like, all right, well, you have the buckets of paint, because they wanted me to do a huge mural over the side of this wall, which they asked me, do you know how to draw murals? And I'm like, sure, absolutely. And so, fake it till you make it, it's a great thing. And so I got to Madagascar, and they're like, so where's your paints? Where are your buckets? And like, I thought you said you had everything. And I was like, no, we don't. And the interpreter, meanwhile, I think was trying to steal my job and tell me, like, oh, no, he doesn't know what he's doing. I don't know why you fired him. And so I'm sitting there like, I don't really know what's happening. And meanwhile, the director of the National Park, the other one, she was like, you're working for me now. Like, this is just a shit show kind of thing. So leave this behind, work with me. And you're going to be drawing lemurs, moths, and birds to make those little plastic pamphlets that you'd see in like, national parks or state parks. And even those are still under construction. We're still creating those 8 years later. But what I like to say, if you're in an internship or anywhere outside of this or the next step ahead, plan ahead, be very communicative with what might be there and what they have and what they don't have, and also just learn how to work independently to make the most of your time in an internship. And I have some of the sketches and illustrations up here if anyone would like to see those after this. But those are some unexpected outcomes. You really just kind of throw you for a loop. You don't really know what's happening. And so I'm in Madagascar, and there I'm like, I just finished like undergraduate in science illustration program almost. I'm like, I want to be a botanist. Like I studied botany. Like, oh, plants are so cool, I love it. And I go to Madagascar and I'm seeing all these endemic species there that are found nowhere else, and like, I'm having time of life. And suddenly, I'm at the table at the research station with a few botanists, and they're talking, like, just arguing intensely about species distributions of the specific genus of plants that I've never heard of, but they're arguing back and forth about how important the research is. And I'm sitting, it's three of us at a table, and I'm sitting there like twiddling my thumbs, like, I have nothing to say, and this is not what I expected to be at a table of botanists. Meanwhile, around the corner, there's the table of infectious disease biologists that are like having a riot, they're like laughing, they're crying, all these, like, they're talking about the research. I'm like, that sounds pretty interesting, but I didn't really put two and two together. So I was like, okay, well, I'll bank that, but I don't really know how I'd get into that field. So I'll just kind of keep that in mind, but that's pretty interesting. And so once I leave Madagascar, my next internship was at the Smithsonian, at the National Museum of Natural History. And so through this internship, I was illustrating tiny little flea beetles that are like about the size of a period at the end of the sentence. And so at the end, and these beetles were all, I get there, and the researcher's like, what's your experience drawing beetles? I'm like, well, let's talk about that. And so a lot of the upcoming weeks was him trying to teach me how to draw these beetles because I had no idea how to go about this. But along this way, looking at these insects for it's like hours at a time, and you do this in class as well, it doesn't have to be insects, but you spend all this time observing anything in your science classes, whether it's like ecology or microbiology or physiology or animal physiology, any one of these classes, you're spending all this time getting to know these forms and these functions so intimately. And it really helps to kind of understand what you're drawing when you start asking yourself these questions. And it's a little feedback loop. Well, the more you start drawing these, and you start asking these questions about what you see and why do you see these forms, and how does that inform the function of this organism in a specific ecosystem. And the great thing about these insects is that when they're collected, no one knows what they interact with, and that's what they feed on, what exactly they do. And we know that there's these species, and so they hired me to illustrate these or reconstruct them. So basically, if you got a beetle and a roadkill beetle, like looking smashed with broken legs and just not looking pretty at all, our job as a scientific illustrator is to reconstruct that and then use that as like a field guide specimen so people would know what to look like, what to look for if they were to see that in the field. But doing this kind of work, and when you're drawing just like what we did, you don't have to create a Mona Lisa to be like happy with something that you just illustrated. It's all about practice and using that as a method to observe the natural world. And I found it incredibly useful because sketches that I did years ago, using the technique of just sketching, just being there present with what you're studying, is incredibly useful, and it does stick with you. You think you might forget it, but then you go back to it and you slowly start remembering all these details. And so one thing I like to recall with this is like you have to be creative with overcoming obstacles because a lot of these specimens, like I said, were just broken or roadkill. So I had to look at species that were related to it and kind of illustrate those, take to the advisor and be like, this is what I think the species might look like based off these broken parts. And then we kind of reconstruct them back together. So it's a good relationship between scientist and artist in terms of them creating a final illustration that's accurate. And so, and also detail-oriented work pays off. And the work that each of you are doing now, all the detail-oriented work, everyone will see it. You don't think that when you're doing the work, can't really imagine that people are ever going to actually notice it or see it, but it does pay off. And people do notice when people put in hard work because of shows. And so that's something that I can't emphasize anymore. It's incredibly important to be very detail-oriented and be kind of efficient with what you do, no matter what it is. And so one of the illustrations that I did during the illustration program was just happened to be illustrating species that people at the Academy of Sciences were studying. And they were studying these specific species that are found over there on Sao Tome and Principe, these two tiny little islands off the Gulf of Guinea, off the west coast of Africa. And so I happened to be connected with some of the researchers there from an acquaintance that I met in the science illustration program. So it happened to just be a single networking event that I went to that this science writer was interviewing people at the Academy of Sciences, and she was like, I want you to illustrate these species for my article. And so it's just a simple networking event that actually landed me the job here at the California Academy of Sciences. And they asked me when they emailed me when I was leaving DC, they're like, you know how to draw coloring books, right? Like, in my head, I'm like, why do people ask me if I know how to draw these things I don't know how to do? But sure. And so suddenly I was on this committee to design this coloring book, and then I kind of find myself in this committee with all these high up curators. I'm like, okay, here we are. And so they're like, what's the first page? And I just kind of go with it. And I think they seem kind of interested in it because I guess I'm still here. And so it's kind of a lot of back and forth between communicating and kind of being aware, in a sense, who you're working with and what they might like and what they might want to see. And so that's also helpful with being observant of who you work with. But then suddenly, once we finish the coloring book, one of the bottoms is like, do you draw plants? I'm like, still, like, why are people asking these questions? And this was one of my first drawings that I did when I was 4 years old. My mom kept the original, but I like to show this as like an example of, you'll get better over time if you practice. So when people ask me, like, do you draw plants? I like to show them the happy flowers. I think that's my brother's and I think I'm probably the one on the left, which is kind of confused. But I tell people, it's like, yeah, practice makes more perfect practice. And there's times when I look at plants and I'm like, I don't know how I'm going to draw that, but I'm going to try it. And so that's how I started my job at the Academy, doing botanical illustration, because right when we finished this coloring book, one of the science illustrators just left. And so she was gone, and he asked me, it's like, well, we need someone to draw plants. Do you want to do it? I'm like, well, I'll take up the challenge. And so I learned a lot about the process of how to use pens to communicate what you see onto a 2D illustration. And during this whole process, there was a lot more reconstruction than I expected. You could see in this photograph here, the tiny leaf that was only like 3 or 4 millimeters long. I had to look at that under a microscope to then look at all these tiny little glandular hairs and these types of hairs, different kinds, that are only found in this species. And still to this day, the researchers and the botanists in the museum don't know the function of these hairs. They don't really know what this plant actually interacts with. They don't even know the full species distribution. And so you might think of all these scientists in the field doing this for decades. All of them are still like, we don't really know what's happening if you go into the details of a lot of these species. And same with, on the left, those are little anthers, little flower parts, and a broken flower on the top left. And so a lot of this was talking with my advisor about what these plants actually look like, how to boil them, like little tips and tricks you know as an illustrator, or kind of figure out over time, and kind of look at the function of these plants just by simply observing them before you start illustrating, because then it'll give you a better idea of what you're working with. And so then it kind of has you thinking, like, what is the goal of scientific illustrator? And then a lot, like the most common question I get is, can you just take a photograph or something? And photographs help, but they don't replace our job as a science illustrator. They help inform us further. And so you think, like, if you look at the reconstruction of a dinosaur or an extinct species, or if you've done dissection in class, if you cut open a rat or like a cow eyeball, I don't know if people dissect these days in class, but if you kind of do that, you kind of see like this off-brown color. It's like the same. So like, how do you determine parts of an eye, or like parts of a frog, and they all just look off gray, off brown. It looks pretty hideous and it smells pretty bad usually. But then as a science illustrator, our job is to kind of dig through this and kind of highlight, like, the heart is pink, like the intestines are red, like the lungs are blue, all that stuff, like highlight these different parts. Not because the goal isn't to create a beautiful scientific illustration, it's simply to create an illustration that serves a purpose, and that purpose is to communicate the scientific concept and to have that concept be engaging for all types of audiences. And so that was kind of my realization working there as a botanical illustrator. And if you have, for those of you that have been to the Academy of Sciences, there's the project lab, where you kind of see the scientists at work. You see all the scientists behind a glass window. Have any of you seen that at the museum? What's kind of like the Jurassic Park-like lab, where you can see like dino DNA and like the scientists working behind a glass wall. And then a lot of people like knock knock on the window, and the great thing about working in there is that you can hear everything that people say on the other side of the wall. So they say some pretty interesting things, and it's pretty great. But I'd work in there, and researchers at the museum hated working there because they would be on display and they'd be watched constantly by little kids or people, where you hear conversation about what people are saying about you. I found it pretty funny working there just to see what people would say. But also, that's when I first started working there. In that museum, I didn't know anybody. I didn't know anyone in San Francisco. And so I was like, how am I going to network in this city, especially in this museum, with hundreds of people in every department? How am I going to meet all these people? It seems so awesome. And I want to meet all these researchers. I was like, well, I guess I'll just put myself on display, because then people are going to see me and they're going to get coffee. And then suddenly enough, people would come into the project lab. You're like, hi, what are you doing? And you know, like kids in the sandbox. You're like, hi, let's be friends. It's like, it was basically just like that. And so one of my friends told me that about some of the microbiologists there at the museum. I'm like, well, that's pretty interesting. Go back to Madagascar. I'm like, wait, that's something that I was interested in. And she was like, you know, there's a master's program through SF State and through the Academy of Sciences. And like, I had no idea about that at all. And she was like, well, you should meet some of the researchers in microbiology. And here I am again, where being a botanist, and that's gone. Everything in Madagascar was like, oh, everything's kind of making sense. And I meet the microbiologist at the museum. And I know that my advisor studies infectious diseases and mosquito-borne viruses like dengue, Zika, and other mammalian viruses like hantavirus. And so I joined the Bennett lab, and I took advantage of opportunities that were at the museum. And so, and another tidbit with I'm realizing with people at the museum that no one there was hired outside the museum. Everyone that has a job there was volunteering for at least 6 months in some kind of position. And so one of the curatorial assistants that works in entomology, and she goes on the necropsies too, she actually volunteered in botany for 6 months before she was hired on in a different department. And so it's all about networking and meeting people and trying to be like the best person that you can be when you're working with people, because no one wants to work with someone that's not a good person to work with. They're just not optimistic, they're just not really present. And so a lot of my time then, outside of botany, was trying to get to know Shannon here in the middle. And so Shannon has a long history of studying infectious diseases. She almost died when she was in college on a study abroad. She was basically infected with 3 types of infectious diseases and malaria and dysentery all at the same time. And she was in this bed. She basically realized that these pathogens infected her with every single possible. And she was like, this is absolutely fascinating. This is pretty interesting to hear her story, because she's on her deathbed, basically. She's like, this is interesting that these small microbes have each found a different way to affect me in very successful ways. She's like, this is fascinating. And she lived. She's around. And she's so passionate about what she studies, these microbes, and now she focuses on like dengue, Zika, and another one called chikungunya. And so each of us kind of focus on different aspects of mosquito-borne viruses. And so this is an illustration I made of dengue virus. When I showed Shannon, she was like, it looks like a ball of chubby baby arms and legs, if you think about it. It's so kind of creepy still, when you think about it. But if you've heard of dengue, it's one of the world's worst emerging infectious diseases. About half the world at risk of getting infected because of the vector, which is the mosquito-borne virus. But it's a very simple virus. There's 10 genes, 3 of which code for like the outer protein layers, and you have the rest, which code for the mechanisms that start hijacking the host cell machinery. And so it's transmitted by Aedes aegypti, and there's 2 species, actually 2 species of mosquito transmitted that are basically invasive equatorially. And so that's what dengue, Zika, West Nile, and yellow fever all look like, because they're sister viruses, which kind of brings about why symptoms can get so bad, which is, I think, why Zika symptoms, a lot of them led to microencephaly, because a lot of prior exposure, a lot of those individuals had prior exposure to dengue or West Nile or previous Zika infection. And so that's kind of what chikungunya virus looks like too. And I found the effectiveness of illustrating these viruses in my research presentations or at conference presentations. You could do these sketches yourself, because I got these models through basically through online. So I kind of illustrated models in a different way to help communicate with specific concepts, like the shapes of these proteins. And so, a little bit about my research is that I am sent these little cryogenic tubes of serum or blood from Berkeley or Nicaragua or Indonesia. I have projects in almost every continent. How I got to that point, that's a good question too, because I realized I was at that point where at the museum, Shannon was like, well, we have a shipment of viruses here for you, can you go get it? And so I go upstairs, just walk along the bridge in the front, and this researcher from Berkeley, and I think looking back at it, she had a lab coat on, which I'm like, why are you driving from Berkeley over here to go to the academy? So I'm gonna go inside, but we have a lab coat on. I don't know why. But she hands me this box of just a styrofoam box, and she's like, don't open it here, go to the lab and you can deal with it then. It's like, all right. I didn't really know what was in it. And so I go to the lab and I open up a box of pure vials of virus. I'm like, this is interesting. How did I get to this point that people are now like, here, have this? And so it's kind of that opportunity. It's like, wow, I guess I've come a lot further than I expected, because these things happen, you're like, oh, and you begin to appreciate how much work you have done in the past to get to where you are, which is something that we all don't do as often as we should. And so we start with these samples of blood or serum, and each one represents a sample taken from an infected individual, or asymptomatic or symptomatic. And from there, we isolate the RNA, we create cDNA, because we want to do whole genome shotgun sequencing, which is basically getting all the RNA and sequencing the entire thing that's found in that sample. In order to do that, because we want to see if there's any co-infections. Like, is that person with dengue, did they really have dengue, or did they have chikungunya, or did they have Zika, or did they have another possible unknown pathogen in their system that we don't know about? And so a lot of work we do too is pathogen discovery. And so we have to, once we do this library prep of all these samples, we put it in the Illuminated sequencer, and days later we get gigabytes of data. And from there, we do this whole programming to kind of align the sequences and see what specific mutations these viruses might have to make certain populations of these viruses more successful over time. And so there's the chubby diagram, dengue. And so these samples are actually from Nicaragua. And so the ones that you can see in the tiny little highlighted rectangle, these are where we've, for instance, seen a specific mutation that allows this group, or this specific escape of these viruses to move on from the rest of the population and be more successful in infecting patients further, which is something we don't really usually see. And then from here, we put them in this phylogenetic framework to see how these samples are related to one another, and we have them based off here to easily see when these samples were collected, when we think they were collected, when they were actually more present in the population. And you can see the C, and the NS2, and the E and the M. These are where we see specific amino acid changes in this population. And so for dengue 1, we saw that these samples from Nicaragua all accrued these specific mutations. And then we're now in the process of doing natural selection studies to see, is that specific mutation on the E protein of success mutation, does that allow that virus to replicate more easily in a mosquito, or replicate more easily in a human? Or does it allow that virus to escape our host immune response? And so we kind of look at this in a more competitive framework to see how dengue 1 may compete with dengue 2. And every year, another thing with these viruses is that you don't see each virus spike every year. Because if we were all to get infected with dengue 1, for instance, if dengue 1 came back next year, our bodies have seen it, and so we're immune. It's the herd immunity. But then dengue 2 can come in because our bodies haven't seen it, and so we can all get infected from there. And then dengue 3 and 4, for instance, it's cyclical. So it's a pretty serious issue in Central and South America. But these viruses are all given the opportunity, because of this dense population in Central America, that because a lot of them they do need to have buckets of water outside to get that fresh water, which unfortunately is a breeding ground for a lot of mosquitoes. And so it's this whole cyclic action. We have to look at the community and say hey, let's try to put screens over the water outside to help protect them from mosquitoes. but also trying to focus on curing these viruses as well, and getting rid of these viruses in the population. And so overall, we look at this, our viruses in this kind of framework to see what kind of mutations they might be accruing. And then we talk to our neighboring lab that helps develop a vaccine, because there isn't a vaccine for dengue or Zika or West Nile. And so we look at the mutations and we look at the entire genome. And there's like, okay, this part of the genome is conserved. We see no mutations happening with these viruses here. But this section of the genome, we see all the mutations happening. So this is the part that's actually under selection due to natural selection. And so we can see that in certain populations, and this is the kind of research that we do here. And so in graduate school, I do a couple presentations on virus discovery over morphology of viruses and what virus diversity. And I did one on marine viruses. I think that's interesting. I kind of escaped from the role of like viruses in the mosquito microbiome and what effect they might have there. And kind of thought about viruses in a more global landscape. Like what roles do viruses have overall? And you might not think about viruses in the ocean. In fact, if you get like a spoonful of oceanic water, it's full of only 400 million microparticles. And a lot of those we don't even know the function of. And so that's a whole huge field that's blowing up right now, is the function of these mysterious viruses that have been there for so long, and we don't really know the function of them. And so that kind of got me into this field of, I was like, well, I know people at the museum are doing whale necropsies, and they study marine animal diseases. I was like, well, this is a unique opportunity for me to kind of start studying marine viruses and to go back to that original idea. It's like, I would like to study large mammals or like marine mammal diseases, but I didn't really think of any opportunity that I had in the future. But then this opportunity happened at the museum where they were like, well, if you want to study marine viruses, you could just be on the whale necropsies with us. And so it's kind of that facial expression, like, do I really want to go about doing this? And then, so if any of you have eaten some pretty gross food in here, so I apologize. You don't have to look, but these are photos from the whale necropsies that we work on. And so if you've heard of the gray whales that have been washing up this year in the Bay Area, so I was on the team to do those first cuts to kind of figure out how those whales washed up and what they actually died of. And so another thing to keep in mind, don't do this on your own because it's incredibly illegal. If you anywhere that you touch or collect even a small sample from a washed up marine mammal or even a bone, you could be fined with upwards of $10,000, just like that. And so, incredibly illegal because they're all protected under the Marine Mammal Protection Act. And so my advice to you is you can talk about the whales, but you have to put this in here because this is something that not everyone is allowed to do. And so, something that was like, found myself in these whales because she was like, my advisor told me, it's like, hey, how we could work on the skull to try to find spear bone that's attached to the lower end of the jaw. This is what this one researcher in Texas wanted, because these little ear bones called the bulla, they house layers of earwax in the whale. And so if a female, for instance, if she's 50, she'll have 50 layers of this earwax in her system. And then you can do a core sample of that earwax, and you can see what years she was pregnant, how stressed she was, and what she actually fed on the most, those specific years. So it's that specific information. Like, I had no idea this is what I was collecting. And so, but I think my advisor told me to go to the school because she just didn't want to deal with being inside the school to try to dig it out. But it's one of those opportunities that if it's something that you do want to research, and if the opportunity does arise, then I highly recommend going after it, no matter how disgusting it might be. And there's me really happy after getting my first Ebola. I had to get a hammer and a chisel and hit it in the right angle and pop it out. It's like, just pops right out of the skull. And if you get to the whale early enough, then the wax hasn't melted, and you can freeze it instantly, and then send it off to the advisor in Texas that you could do the core sampling of the earwax, or the ear plug. And so that's my advisor. No Flannery and no blue whale that washed up last year in Bolinas. And so you kind of, when you're in these positions, it's such kind of like an honor to be working with these huge whales that they don't wash up that often, thankfully. but you're in this, you're like, how can I make the most of this opportunity? And you start asking yourself a lot of questions, like why, how did I get here? What can I do to optimize this time for the team and also myself? And think about like, what can I do before I start, for like the plants for instance, before I start drawing, I'm spending a lot of time just looking at them and observing them and asking questions before I dive into it. Because you don't want to go into something unprepared. You want to ask yourself, just mentally prepare yourself a little bit before you start. And the same thing happens with these, where you're walking around the whale. It's honestly smells pretty awful. It's not the best. But you're taking measurements, you're taking note of what you might see on the whale, and you're going to see like, how am I actually going to answer these questions that I have, like how did it die? And how did it get here? And what samples can I take from this whale to then give me the evidence that I need to show that it did die from a ship strike, or from starvation, or from parasites, for instance. And so, and what I highly recommend is everything you just do, even after a class, a lab, just sit and think about what you just did. Was it interesting? Did you like it? Was there anything that you could have done, or the teacher could have done to make that situation better? But kind of think about observe, how do you feel about it? Because that's something that we don't do too often as well, especially in the sciences. And something that I learned about starting these necropsies was the parasites. These are whale lice. So they get about like that big on the whale, and they're nasty because they'll just bite through a rubber glove. These huge like sap-sucking kind of pincers that just like really aggressive and not the best things to see. But I found it interesting because studying mosquitoes, like what are the function of these whale lice overall? I don't know of any researchers that are actually studying them. And I have some people in our lab that are studying the mosquito gut microbiome. For instance, like what kind of bacteria are present in the mosquito gut that when they're present, how come it prevents the transmission of dengue or Zika. So there's like gut microbiome competition happening in the mosquito to prevent transmission of Zika or dengue. Can that happen within these whale lice? Do they transmit viruses? We don't know. No one has really done microbiome studies of what these things might have. And also that the barnacles are species specific, which is interesting to the whales. So each whale has a very unique set of barnacle species that they pick up when they go to certain sites to breed. And so I started collecting all these barnacles and these lice. I haven't done anything with them yet. I've just gone on these necropsies to collect them, because maybe in 10, 20, 30 years I'll go back to the museum and probably hopefully study them, or if not, then someone else will. And so there's these very open areas of research that in undergrad, you kind of have this thing of like, oh, everything is known. There's so much is known about the world, like everything's been studied. So like the plants that I work on, those are all new species. The beetles that I illustrated, those are all new species. People don't know anything about those. For all we know, some of these, this might not even be one whale louse species. It could be multiple ones. And the fact that they have a social behavior, kind of like ants, which we realized when we were like just looking at them feed, we're like, this is pretty interesting. Like, how can this relate? If they were a vector, how could this relate to other whale species? Like, can they be a transmission of unknown viruses or pathogens? And so just something to think about when you do get in situations, just like sit back and observe and kind of look at things overall before you start diving in. And so, again, the disclaimer, you see a dead whale on the beach, you can call it marine mammal center, but don't try to get fresh with it, don't take anything from it. Highly illegal. And so, another thing what we realized is that if you're in the whale, this coworker took a photo of me, so I was in the skull, and I popped the tongue, and brown gas started flowing out, just spewing everywhere, and I kind of got dizzy, because it's a lot of methane that's like popping out of it, so you kind of just have to be present with what you're doing. A lot of times, even if it's not the prettiest going to work, but you have to keep a good attitude, because I know over the years of working in the museum, I've met some of the most intelligent people that I've met, but they've been not the best people to work with. They're not happy to work with you, or they're just not they're not there to socialize, they're just there to work, and that's it. Or they're just really just not the nicest kind of people. And so they could be geniuses and kind of figure out a cure for cancer. But people won't really want to work with them because it's not pleasant to. And so that's why I like to say keep a good attitude, because those of us that are working on the whale necropsies, kind of have to joke around and laugh around a lot because you're in something that's so grim. You have to keep a very positive attitude. And so, you like have that, this is fine attitude, because usually sometimes it's not. And so, like I said, it's very unexpected how all these things happen, but a lot of it is just being aware of your surroundings and what might be presented to you. And so these are kind of different methods that I wish someone told me to kind of be a little bit more aware of research in STEM or opportunities in STEM, or how to, even if the opportunity isn't that obvious, to kind of just like look around yourself and see. And one of the things that I highly recommend overall, and most important, is to volunteer in a network, because that is going to get you connections that are going to provide these opportunities for you in the future. And obviously keep a good attitude because you want to work with great people. But being observant of what you like and what you don't like are equally as important as well. And then you want to find ways to align your interests with your goals and find that time to kind of look at what you're interested in and how can that tie in with the goal that you want. Do I have a goal? Like I'm applying for PhDs right now, but do I really know what I want to study? It's like, I don't have a specific project in mind, but I do want to study green animal diseases. But do I know the different route that I'm going to take to get there? Not specifically. And so that's kind of the challenge right now for me in STEM, is to take that next step and to figure out where that's going to take me and where that's going to lead. But that's kind of the whole overall story with these people that go like lecturing at you, talking at you. It's like, I've been in this field for 40 years. Like, that's not true. Like, most of the time, it's like, how did you find that way? Like kind of like when you see a finished painting, like you don't see the hours of sketches and the conceptual time that went into that person developing and how they have a passion for that field. You kind of just see the final product up there. And so that's something to keep in mind, like the amount of hours that takes anyone to kind of get to where they are, within like years, for instance. And so that's kind of one of the most important things to keep in mind. And if you don't really know, then I recommend creating one for yourself. And so a colleague and I at the Academy of Sciences, we started 5 career scientists because those of us in the LGBT community that don't really have that outlet to talk about a research or to be present, it's very limited. And so if you've heard of 500 women scientists, we kind of model our organization after the same with their permission. And so we kind of give this platform for people all over the world that do identify as LGBT to kind of submit their stories online, and we post them on Instagram and highlight 3 to 5 different researchers every year about who they are, how they identify, what they study, why they study it, and what does it mean to be LGBT in their community. Because a lot of times, these researchers might be in a complete opposite situation in San Francisco, where you're in a situation you can't be out, you can't be open, you can't be yourself, and that does affect your workflow, or who you are, and how happy you can be, what you can actually do. And so it was important for Lawrence and Zito and I to create this home for those of us in the community to kind of share who we are, and kind of own who we are, and share that with the world. And so I was able to do that too as a freelance illustrator, as studio names and pangolin, because I focus on endangered and threatened species, because like the pangolin, a lot of people don't know that it exists. People like, cute, an armadillo. They're not too related to armadillos, but I can get into that. I could talk about these guys for days. But I want to create a studio to kind of focus on endangered and threatened species, to kind of show people that these species are in trouble and we need to give them help as much as possible, because we don't know if they're knocked down from an ecosystem, what effect might it have on the whole trophic system and the ecosystem overall. And so I've found it very useful to use illustration for community purposes and for research. And so, and I could tell you more about the other illustrators in the program, what they focus on and what they do, because we all focus on something very different. It's been amazing to see where people have gone from here. And then other opportunities you can have through the sciences and arts, combining the two, are joining these art or science residency programs. I just heard about science residency programs a couple of weeks ago. I didn't know that was a thing. But basically these institutions will kind of hire you, give you money to go there and just study for like 2 weeks or a month. or just go out into their field and just be there and just ask questions, write it down, and you'll be able to share that with the cohort that you're with. The same thing with these art programs, the art residencies. A lot of the national parks, of the 59, I'd say over 50 have art residencies. And if you have a creative outlet, no matter what it may be, you can join these art residencies and apply for them and spend 2 weeks or a month in a specific national park, all to yourself or with your partner, and just kind of immerse yourself in nature. And I know in Crater Lake, when I did the residency earlier this year, they hired a plein air painter, and then me, an illustrator, and the artists that are coming later this semester. One is a writer, and one is a kind of nature-based comedian, where they're part of Second City Network. And so they kind of go out into specific parts of national parks, like, and make jokes about Half Dome, or they make these videos about it. And so, like, you could really, literally do anything and apply for these art residencies. And it's a great way to try to figure things out, because we're all still trying to do that. And so if anyone has any questions, I think my advisors at the museum, in the lab, yeah, and if anyone has any questions, feel free to email me, message me, and open up to questions if we have time. Thank you. Does anyone have any questions? Yeah.
So, as an artist, how do you approach, like, what are your thoughts on the emergence and the growing popularity of, like, digital art for scientific purposes? And do you feel like there is, like, an advantage to still using, like, traditional art mediums for scientific illustration?
That's a great question. During the illustration program, they taught us part-time digital techniques and part-time traditional, because they wanted to like throw you out of the program, being well prepared, having your own business, like kind of being able to tackle, like, if someone has to draw a butterfly in watercolor or a tiger in Photoshop, and be able to like teach you how to do that. And so I found that the digital hasn't replaced traditional at all, that you could do computer-based illustration. I have some friends that strictly just do digital. And it did show during the program, some of us focused on graphite or penning or painting, others just did digital. And it's kind of the medium that you feel most comfortable with. But I feel like they have not replaced each other, thankfully, because I feel more comfortable with digital. But some of the work I had up here was digital, and it's fun to like get to know it. Obviously, it's a challenge to like tackle something that's unfamiliar and to get your comfort zone, which is another thing I highly emphasize. Like, don't stay in your comfort zone. It's not really moving that much. But yeah, I feel like they are both equally as important.
Just on average, how long did it take you, like, when you showed those leaves with the hair, like, how long does, like, illustration like that take?
A great question. When I first started there at the museum, trying to get, what took me the longest was getting to know my advisor and what he liked. Like, he noticed, like, when he said, like, oh, I really liked when this illustrator did that. I'm like, can I take note? Okay, can I take note of what the advisor likes and what they want? But then learning to do all the small details that, at first, a whole plate, a typical plate would take me about a month to do that, but now getting to know the advisor, the pens, and what they like and what they don't like, it takes me about a week to do each one. And so kind of get to just popping them out after a while. But yeah, sometimes I have clients that look like little teddy bears that are just completely covered in hair, and like, well, it's going to take a little longer than I expected, but you know, it depends on the amount of detail.
When you were still in your undergrad, you were like hunting for like volunteer opportunities and like internships. How did you like approach like going for them or like getting in touch with a bunch of like researchers, advisors? You still keep in touch with old advisors that you did back in your undergrad?
Yeah, I still keep in touch with advisors that I, even after the semester started, you know, yeah, from my office hours, if you have any questions, I would just go and meet with them, because if it was paleontology or if it was botany, or being a botanist, I was like, I have some questions, like, I I'd love to work with you. I would basically just go to their office hours and just meet them and then go like a few times a semester just so they will recognize me on a first name basis. And a lot of those advisors were able to write me the letters of record that I needed to get into the illustration program. And one of them wrote me a letter for the master's program at SO State about almost 8 years after. And so it was an ongoing connection with them. And meeting them like, hey, hope everything's going well. I did this one thing, thought of your research, hope everything's going well, kind of thing. Just have that simple connection with them. They do appreciate that. Everyone does really love to hear back from people that we interacted with a long time ago. Of course, most of the time these advisors will like thousands of emails, so it's like, so most of them won't get the response, but be annoying, email them again, show up again, because advisors, we all just forget emails, and we all do it. But I recommend just showing up to class, and even after class, if you have a chance to ask them some question about how they got there, or anything about what they just talked about, I would recommend any opportunity to get to know them on a real personal level. Because if it's a lecture hall, like 40 to like hundreds of students, how are you going to do the work you need to do to kind of show up and to kind of be more present? Be a person that they know and recognize. So even like simple connections of like going up to say hi, it seems not scary, but it's something that they like to see.
Is there like a threat to kind of job demand for this? Because like now with all the AI and computer graphics and stuff, is that beginning to come in, or would you say that like science illustration is like a pretty stable and it will be even after pictures and everything?
Yeah, another great question. I found that as long as there's science, there will be a need for illustration for someone to communicate it. And so one of my friends actually works with biotech in San Francisco, more as a science writer, as a science illustrator, but they have such complicated machinery that they're dealing with, and what they do with all this data and samples that they collect, and then kind of making that very simple for patients to understand. A lot of scientists are like so stuck in research, they're like, they don't understand why they don't understand like genomics and things. This is so simple. And so they need to hire these people to either illustrate what they study or to rewrite it to make it simple and approachable for everyone else. And so, yeah, as long as there's science and research going on, there's going to be need for illustration. And I haven't found like the digital world hasn't replaced illustration at all. And so, one thing that I have done is kind of like forging a pathway to like via networking and kind of be present and to kind of focus on endangered and threatened species. Like that's my thing now that I focus on. And I have some illustrator friends that focus on like dinosaurs, some people that focus on classical anthropology, so they focus on like broken pottery and reconstructing that, or like bow and arrow, for instance. They'll be creating these illustrations for museums, or like huge canoes from Fiji from hundreds of years ago. They'll be at the Smithsonian in the basement illustrating these canoes that they can only have access to at certain positions. So it's kind of crazy to see what these people are actually illustrating. But yeah, as long as there's someone stepping up, there's going to be need for other people, illustrators or writers, to communicate that to the public. Yeah. If there's any other questions, if not, there's a little refreshments over here too. Thank you, everyone.